Assalamu alaikum. This is the tutor from Inlaw Online Academy, and we are going to solve biology IGCSE Biology paper for 2021, and it's paper for May June 2021. Okay, let's get started. All right. So the first question here says that Baker's yeast Saccharomyces cerevisiae is a single-celled organism that is classified in the kingdom fungi, and we have got a diagram for it. Then they're asking state one other kingdom that contain organisms that all have structure A. So structure A mainly tells you about the cell wall. Okay. Now this is yeast. Yeast cell is from kingdom fungi, right? And it has got cell wall. Now the other kingdoms that contain organisms containing cell walls in the cellular structure, those are plants and sometimes prokaryotes such as bacteria. So plants. Okay. Then Table 1.1 shows some cell functions by naming the cell structure responsible for each cell function and give the letter that identifies each cell structure. Now, cell function is storage of genes and the cellular structure. Now, let's talk about fungal cells. The fungal cells are eukaryotic cells. And eukaryotic cells are going to have a well-defined nucleus, and their genetic material or their genes are going to be in the inside the nucleus. ठीक है DNA on DNA. ठीक है DNA के ऊपर inside the nucleus DNA is their genetic material is stored in the nucleus. It is present in the nucleus. So cell structure, that would be nucleus. And where letter from the diagram that is mentioned above, let's see. So it would be now this. Okay, so it's G. Okay, you can see here. Okay. So letter is G. Then aerobic respiration and the cellular structure. So aerobic respiration of yeast takes place in mitochondria. Aerobic respiration takes place in the presence of oxygen. And the letter that represents mitochondria here is E. Then amino acids are assembled to make proteins, and remember that amino acids assemble to make proteins are a cellular structure that is responsible here for sure. That can be rough endoplasmic reticulum, as ribosomes are present on the rough endoplasmic reticulum, and ribosomes mainly are responsible for transcription of the translation of the transcribed genetic code that is present in the messenger RNA into the amino acid sequence that is then assembled to make the proteins. Okay, so that way you can say ribosomes or rough endoplasmic reticulum. It depends. We can write any of these. Ribosome, and the cellular structure could be okay. So it can be okay. So it can be H, which is like these ribosomes. And it can be F as well. Like this is rough endoplasmic reticulum. Depends. If you have written as a rough endoplasmic reticulum, then it would be F, and if you write ribosome, then it would be H. Student made a drawing of one Escherichia coli bacterium, and that has been shown. So actual length of the bacteria cell is 2 micrometer, and we need to convert the actual length of the cell to millimeter. So actual length is 2 micrometer. You want to convert it into millimeter. When we convert micrometer into millimeter, we need to divide that value by 1000. So the value that we would get would be 0.002 millimeter. State other information that the student needs in order to calculate the magnification of the drawing. So the formula for magnification is um, size of the image divided by the actual image. Okay. So actual length is given. The student needs the size of the image. So it would be size of the image, because magnification is equal to magnification equals to actual, sorry, size of the image divided by actual length. Okay, then describe the similarity and differences between the structure of yeast cell and the structure of bacterial cell. So when we are to talk about similarities, the yeast cell and bacterial cell both have a cell wall and cell membrane. Both have cell wall and cell membrane. You can take help of the diagrams mentioned above as well. Cell wall and cell membrane. With that, I'm mentioning the similarities first. You can write like maximum 3 to 4 similarities and 3 differences, as the question is 6 marks, so at least 6 points should be. Do not write in the form of points, you know that, right? So at least 6 lines you should write. So both have cell wall, cell membrane, they have got cytoplasm, and they have both of them will have ribosomes, and yeah, they are unicellular, both of them are unicellular. Okay. And if we are to talk about the differences, so differences we can say, now yeast cell eukaryotic is a eukaryotic cell. That means that they have got well defined nucleus, or their genetic material is enclosed in nucleus. And bacterial cell is a prokaryotic cell, and their genetic material floats in the cytoplasm. Then that simply means that yeast cell is a eukaryotic cell, so it would contain a well-defined nucleus. Bacterial cell won't have a nucleus. With that, bacterial cell does not have a mitochondria, does not have any mitochondria, no mitochondria. Bacterial cell, while yeast cell has mitochondria, and the aerobic respiration of the yeast takes place in mitochondria. Bacterial cell contains plasmids, but yeast cell does not. With that, yeast cell has a large vacuole, while bacterial cell does not have any vacuole. Then some bacteria are involved in nitrogen cycle, and the nitrogen cycle has been shown right in front of you. So first is that they are showing you the conversion of nitrogen. First step, see the first step. It is nitrogen being converted into ammonium ions. Acha. So this happens in the bacteria. Okay, it is mainly nitrogen fixation, and the nitrogen fixing bacteria are found in the root nodules of some plants. And we know that atmospheric nitrogen cannot be really used by plants directly, right? It needs to get converted into the usable forms, which could be in the form of some nitrate or nitrite ions or ammonium ions, or when we talk about fertilizers, the fertilizers actually contain nitrogen in the form of ammonium salts, or yeah, salts like that. So nitrogen fixing bacteria, what do? They fix nitrogen, atmospheric nitrogen, into the usable ammonium ions. So first is nitrogen being converted into ammonium ions, so that is nitrogen fixation. Then second is proteins being converted into amino acids, and then amino acids being converted into ammonium ions. Now this is deamination. Why is it deamination? As deamination is actually breakdown of amino acids into ammonia, and that ammonia can be then used in many other ways. Ammonium ions make conversion of the air. Like if we talk about human body, right now it's not really about human body, but when we talk about human body, so in human body deamination is actually happens when like proteins are broken down. We consume proteins are broken down into amino acids, and then amino acids are actually broken down into ammonia, and then ammonia is then removed from our body through urine. So that's about human body, but here in nitrogen cycle or when bacteria are involved, so it is mainly breakdown of proteins into amino acids, then amino acids into the ammonium ions, and then those ammonium ions or ammonia containing salts, if they are formed, then they can be used by plants as well later on, or when they are converted into the nitrate and nitrite ions as well. So deamination. Because in deamination, the amino group of amino acids is actually removed, and that amino group is then converted into ammonia. Okay. Then a third point is actually showing you um, conversion of ammonium ions into nitrate and nitrites. Now that is nitrification. Nitrification. Carried out by nitrifying bacteria. Okay, then question number two is large quantities of plastic waste are polluting the oceans. A survey published in March 2018 showed the increase in plastic waste in Pacific Ocean. One area of the Pacific Ocean is known as the Great Pacific Garbage Patch. Data were collected from areas inside and outside the Great Pacific Garbage garbage patch, which is also represented as GPGP, between 1965 and 2015 to estimate the quantity of plastic with. Now remember that we don't really, for your convenience, I've read the question, but we don't really need to read the question throughout. Like, it's not really necessary. You don't really need to waste your time reading the question. You can simply move forward towards reading the graph, and it would help you out either way, like reading the graph. And if you don't understand still reading the graph, then you can go towards reading the question. But most of the time we can understand the graph on its own. So here on y-axis, we can see that the estimated quantity of plastic waste per kilogram per kilometer square of ocean surface has been shown, and then the years on axis have been shown. The shaded, dark shaded is inside GP. The plastic weight, quantity of plastic weight inside the Great Pacific Garbage Patch, and then the lightly shaded region is outside. So we need to describe the results of the survey shown in figure or the graph. So if we talk about inside a Great Pacific Garbage Patch first, so inside the quantity of plastic weight inside the Great Pacific Garbage Patch is always higher than the outside. You need to take an account of the sea. Okay, it's always higher throughout the graph. So first thing is that inside always higher. Okay, inside always higher. Then um if we have to like see or if we are to compare, just a minute. Okay, so from 1965 to 1965, to 1974, 1975 to 1984, and till approximately 1985 to 1994, right? So the quantity of plastic waste inside the GPGP, okay, that is constant. So you can say from 1965, 1965 to any of the 1984 or 1918, 1994, you can say, or 1984, that is constant, right? The quantity of plastic waste. Okay, so let's talk about outside. So the increase in the plastic waste outside the GPGB, that actually fluctuates. Like it increases from 1985 to 1994, right? And then it decreases and then it increases again, right? So it's kind of fluctuating throughout. And inside GPGP, it's like um, it's there's a steep increase. It's constant at first and then steep increase. It's increasing. Okay, so that's kind of the main things, main points that we are going to note right here. Then we're going to move forward and talk about part B, which is the green turtle. A turtle, Calnoia midas, is a species of marine animal that is harmed by plastic waste, and it shows a green turtle swimming past a plastic bag in the Pacific Ocean. Okay, so turtles are classified as reptiles. State one feature shown by reptiles that is not found in amphibians. Now remember that कि reptiles जो होते हैं, they have got a scaly skin, and amphibians will have that slimy skin, right? Then reptiles lay their eggs in on land, and जो amphibians होते हैं, they lay their eggs in water. So you can mention any of these. So um, reptiles lay their eggs on land. Okay, just reptiles. Outline the dangers of non-biodegradable plastic to marine animals such as green turtles. Now, non-biodegradable plastic is the one that does not break down. Okay, and it's not really going to, like, if the marine animals digest this plastic, it's not really going to get digested as well and excreted out as well, right? So it can lead to toxicity. Okay, first thing, if injection takes place, it might cause toxicity. Then it might cause suffocation as well. Ingestion of it might cause suffocation as well, like a large piece of plastic. They ingest a large piece of plastic, it might cause suffocation and then death. And ingestion ingestion leads to toxicity, suffocation. Then they might get trapped. Trapped in the plastic, and that might cause damage and serious injuries to them. Then plastic, if we talk about indirect effects, so it would be like when plastic gets accumulated on the surface, okay, of water. Then what would happen is that it might block the sunlight, and water, it might block sunlight. Plants and algae, marine plants and algae won't be able to photosynthesize. No food, they might start dying. And with that, food chain would get disturbed. With that, plastic accumulation, in the sense that if a fish ingests the non-degradable plastics, the plastic, so what would happen is that if there is another fish that ingests that fish, okay, so that non-degradable plastic is going to get accumulated now, and it's going to get accumulated in larger amounts there. Then that's how the plastics are going to get increasingly high throughout the cycle of ingestions. Okay, so plastic accumulation by the accumulated up the food chain, and that is bioaccumulation. If you're familiar with the topic of bioaccumulation, you would be able to understand it. Then suggest ways to reduce the quantity of plastic waste. Now what we can do is that we can recycle and reuse the plastic. Less plastic, less new plastic produced, plastic manufactured. Awareness campaigns for reducing the use of plastic. with using biodegradable plastic. These can be the measures in order to like reduce the quantity of non-biodegradable plastic waste. Then question number three is bacteria are used in many biotechnological processes, and explain why bacteria are useful in biotechnology. Now, bacteria are very well studied organisms. Firstly, then they do not require much space. They're very small, do not require much space. Now, studying bacteria does not have any ethical concern. If we would study like a human cell or an animal cell, and we are trying to grow that out, and we want to develop it, we want to see the reproduction and develop into complete offspring or a baby, so that would have quite many ethical concerns related to it. So there is no ethical concern here. And then bacteria grow at a very rapid rate, so they don't really require a lot of time for growth. So for that reason, we also prefer bacteria. Rapid reproduction rate. Then plasmid is a characteristic of bacteria. That is, like, bacteria has got a DNA, okay, a coiled DNA structure. But in addition to that, bacteria also have plasmid, or plasmids are really great vectors when we talk about genetic engineering. So plasmids are available, which are very great vectors in genetic engineering. Okay, then insulin is one of many human proteins that are made by genetically engineered bacteria. Some people cannot produce insulin because their immune system has destroyed the cells that make insulin. Now we need to state the organ that contains the cells have been destroyed. Now that organ would be pancreas. Pancreas is the organ that actually produces the hormones which regulate the blood sugar level, and those hormones are insulin and glucagon. So these cells have been destroyed. The cells in the pancreas would be destroyed that way. And state the name of the disease caused by destruction of the cell of these cells. That is type 1 diabetes. And then state the function of insulin in the body. Now insulin is a hormone, okay, which is generated or which is secreted by the pancreas. When it is secreted, when we consume our food, sugar containing food, our blood glucose levels rise. And when the blood glucose levels rise, the insulin hormone would be produced by, or would be released by, the pancreas. And insulin hormone is going to see the blood levels, and it's going to try to maintain them. And how it would do that, it would take the glucose out of the blood to the cells, and in the cells the glucose is going to get consumed. So by taking out the glucose from the blood, it actually lowered the blood glucose level, or mainly maintain it. So lowers the blood glucose levels in order to maintain it. Genetically engineered bacteria that are used to make insulin when grown in a fermenter for 5 days. Samples were taken from the fermenter every 6 hours, and the number of bacteria in 1.0 millimeter cube of the nutrient solution were counted. Now, changes in the number of living bacteria in the samples taken from the fermenter are shown. Okay, so 1 and 2. We need to add the labels x axis and y axis. Now y axis पे क्या चीज shown है, y axis पे हमें time shown है. Time in hours or days depends, you can mention accordingly. And y axis पे population growth of bacteria, or number of bacteria. State the names of the stages of the population growth of bacteria. So bacterial cells, population growth of bacterial cells, so that is P is lag phase, ठीक है। लैग फेज क्या होता है? लैग फेज में जो बैक्टीरिया सेल होता है दैट एक्चुअली ग्रोज इन साइज एंड द पॉपुलेशन ऑफ बैक्टीरिया इज नॉट रियली गोइंग टू ग्रो इन द लैग फेज. Log phase is the phase of exponential growth, and in this phase, what happens is that the population of bacteria is actually going to grow now, and R is like the stationary phase, and in this phase, the growth of bacteria is going to stop. Then death phase, the bacteria die. Explain with reference to figure 3.1 why the bacteria did not grow in the fermenter for longer than 5 days. Now, for longer than 5 days, why did the bacteria does not grow? That would be the death phase being there, okay? Or why would that happen? That would happen due to that there can be many reasons to it, okay? Maybe the nutrients that you had provided to the bacteria would no longer be available. Now, due to overpopulation, okay, with the passage of time, the growth of bacteria is also going to increase. And as the growth of bacteria or the population of bacteria actually increases, there would be a lot of competition. That competition leads to competition for space as well. So no more space available. There could be a reason that no more space is available and the bacteria die for that reason. No more space for space available, and then the competition for nutrients as well is there. So no more nutrients available, that the nutrients have been used up, that you provided to the bacteria, and the pH might change and the acidity increases, or the waste that was being produced by the bacteria that can kind of get built up. So buildup of toxic waste substances might lead to the death of the bacteria. And not enough oxygen available as well, because there are a lot of bacteria consuming oxygen or respiring aerobically and taking in oxygen. So that takes place. So these could be the reasons, possible reasons. Then mineral salts are important in the human diet. One of the most important is iron. Explain the importance of iron in the human diet, the effects of iron deficiency. Now, when we talk about iron, so iron is a mineral which actually is involved in the formation of the heme portion of the hemoglobin of blood. Now, if we talk about the heme group, that उसका सेंट्रल एटम जो होता है ना दैट इज आयरन. फिर उसके साथ ये ऐसे नाइट्रोजन जो होते हैं वो अटैच्ड होते हैं, एंड देन अदर स्ट्रक्चर्स आर अटैच्ड विद, बट सेंट्रल जो एटम है वो नाइट्रोजन है. ठीक है. सो फॉर दैट रीजन when we talk about importance of iron in the human diet, so it's really necessary for the formation of the red blood cells of your blood, is involved in the formation of heme portion, formation of hemoglobin. All right, hemoglobin. So um hemoglobin of blood. And remember that hemoglobin that is present in the red blood cells, okay, hemoglobin is involved in like binding to the oxygen and then carrying it to the cells of the body where it is required. All right. So if iron deficiency takes place, it actually causes anemia. Now, it is a condition in which body would not have enough healthy red blood cells, right? And इस condition में क्या होता है कि a person starts getting really pale. The person's eyes are going to get pale. Now, as the red blood cells are not healthy enough, so red blood cells for sure are involved in the providing oxygen to the body. And when red blood cells are not healthy, anemia is there, iron deficiency is there. So for that reason, what would happen is that enough oxygen would not be provided provided to the body. And for that reason, the symptoms that that person is going to have is like dizziness. He won't really have energy, because oxygen is really an important part of your respiration, and respiration is the process through which your body gets energy, right? So energy nahi hogi, and the person would feel less energetic and would feel really dizzy. And yeah, so that's all about it. Then shows figure actually shows you a field of cassava, and we call it many hot esculenta, which is a crop, a plant grown in parts of Africa and Asia. The plant stores starch in their roots, which form a large part of the diet for many people. Cassava does not provide many vitamins or minerals, and genetic engineers have modified cassava to increase its iron content. They have done this by incorporating a gene from a membrane plant, from a membrane protein from the plant Arabidopsis thaliana. Now state the name of the enzyme that is used to cut out the gene from the DNA of A. thaliana. That is a restriction enzyme. Okay. Now describe how the gene from A. thaliana and the DNA from cassava form recombinant DNA. Remember that the gene from the A. thaliana has been cut by the restriction enzyme. Consider these the segments, okay, of the genes which have been cut from the A. thaliana. Now the DNA from cassava, ठीक would be taken. The DNA, it is circular DNA. Now क्या किया जाएगा? The restriction, same restriction enzyme which was used to cut the A. thaliana gene, to get the gene from the A. thaliana, is going to be used to cut DNA from cassava at specific points, to at specific points, ठीक है? Restriction enzyme would do that. फिर क्या होगा? Sticky, this way sticky ends would be generated. Sticky ends would be generated. So this is the DNA that has been cut. Sticky ends generated. Now what happens is that when we mix these two together, the complementary ends and add ligase as well. The complementary ends are going to bind together, and the DNA that you got now is a recombinant DNA which contains the gene from A. thaliana. So same restriction enzyme is used to cut the DNA from cassava, which was used to which was used to get the genes from Italiana, and then as a result of which sticky ends are generated. Sticky ends are generated when the fragments, both of the fragments are mixed together. Those that would be complementary to each other, or the complementary ends, are going to bind to each other. The complementary ends bind to each other, each other and sorry complementary ends bind to each other, and complementary ends bind to each other, and that is by the addition of ligase, and by adding ligase binding takes place, because ligase is the one that actually binds them together, takes place. You can change the wording. Most suitable wording is the most suitable wording. I actually added ligase at the end. You should add while the um, binding takes place right before that. So suggest who developed genetically engineered varieties of crop plants, often breed them for several generations before releasing them for farmers to use. So suggest why the scientists do this. Now there could be many reasons for that again. Okay, scientists, why do they do this? To be safe. Okay, so that they can check, okay, if the offsprings are going to inherit the new trait, or to ensure the new variety. So offsprings inherit, to check if the offsprings inherit the new trait, or are the new genes do not have any adverse effects, like the new genes are causing any disease in the plant or anything like that, so do not have any adverse effects. Or the new variety is giving is maintaining the high yield that was for the plant it before it was engineered. So new population, high yield is maintained, and to ensure that it actually can grow in field condition, field conditions. All right. So that would be it for this question, and that would be it for this part. This is the first part of paper 4, June 2021. I'll be explaining the rest questions in the next part. So that would be it for today, and see you in the next part. And goodbye. Take care. Allah Hafiz.